Okay, good morning, everyone. It's actually a great honor for me to have this opportunity to give a talk on metal adventures from Earth to life. I'm Jiubin Chen, as just showed. And today I will talk about why focusing on metal and how to use stable isotopes tracing metal adventures from Earth to life. So first of all, I would like Dr. Sir Paul Gast, and actually I think he was born in Chicago, and without him, I won't be here to give this talk. And also, I would like to the jury members of my PhD defending. You can see in this picture, especially my supervisor, Dr. Jerome Gayade, the guy with the beautiful tie, and the only one. And also Tom Bolin with a blue shirt. Without their permission, I won't start my adventure on metal stable isotopes. And also my students who always work hard, and definitely they deserve a beer, as you can see in the pictures. And all bigger thanks also to my colleagues and friends, and who support and help me all the way along. So before I start my talk, I would like to convince you that the adventures of metals from Earth to life is a story-filled journey. So as we can see from these pictures, then the environment and the life for the past 4.6 billions didn't go separate ways, but instead co-evolved throughout the history of the Earth, as reflected here, for example, by the similar variation of oxygen on the environmental side on the top, the red curve, and also the biomass volume on the life side on the other side. Why and how this happened has been a mystery, but may be encoded in the journey I'm about to tell you. So this is because the elements of both major and micronutrients as information and material carriers witness this co-evolution. And in this regard, conventional stable isotope studies of nitrogen, carbon, sulfur, and also oxygen have made great headways. But the metal side of the story has largely not been told yet. In fact, metal as micronutrients play a pivotal role in writing the history of life environmental co-evolution, and more importantly, we know metals are not degradable in the environment. And the life can only obtain metals from the Earth's environment, and the journey of metals from and back to the environment through life would record everything about the co-evolution. So the correlation of life and metal is not a secret, as we can see in this on the left. Nanothemes image where the spatial distribution of iron overlaps with that of carbon and nitrogen in the fungal cells. And for Earthlings like you and me, you may or may not be surprised to see that the metal distribution in our blood is very similar to that of our crust. This suggesting that we humans are probably the product of this co-evolution. So, in fact, the metals actually act usually as the active centers of protein and enzymes. For example, 35 to 59% of enzymes are metalloproteins. And the functions of metals in life are not something that can be easily replaced, because they play activities only they can do due to their unique electronic structures. And because of this, metals can affect a range of bioactivities, including, for example, modifying metabolic pathways, inducing gene mutations, and promoting protein synthesis. So in fact, the availability of life essential metals have on one side echoed with the environment changes such as GOE, and may have directly influenced metabolic innovation and biological evolution. And the correlations of metal global availability in Asian Ocean and the key geological events like GOE or NOE, and also the emergence of metal proteins are shown in these three figures. And together, we can see that in geological history, the emergence of evolution of various metabolic pathways are correlated very well with metal availability. And give several examples. For example, before GOE, the manganese binding protein promoted oxygenic photosynthesis. And after GOE, molybdenum-related proteins constrained the evolution of eukaryotes by, for example, nitrogen fixation. And the copper zinc-related proteins, together with other metal proteins, also promoted advanced life and even the Cambrian explosion. And most recent research even suggests that metal stress might cause the evolution from prokaryotes to eukaryotes. And the key to assess the impact of environmental metals on life evolution is to understand how environmental metal into living organisms and play their roles. So this is not only necessary to clarify the concentration, the speciation, and structure of metals in either environment or living organism, but more importantly, to accurately describe the dynamic processes such as the environmental cycling of metals, their transport from environment to living organisms, and also finally their metabolic cycling. So one of the best tools to track the dynamic processes of metals in both the environment and the living organism is stable isotopes. So in this talk, we focus only on three metals as copper, zinc, and mercury. So their mass dependent fractionation is expressed using the low-case dirt, and I want to mention that for mercury, there is also mass independent fractionation of both all the isotopes capital delta 19921 and also even isotopes capital delta 224. And all the MF is thought to be generally caused by either nuclear volume effect (NVE) or magnetic isotopic effect (MIE). And up to now, the mechanism for even MF remains still unknown. But I just want to mention, as you see, that the mercury is the only known element that has a unique three-dimensional isotopic fractionation pattern, mass-dependent fractionation, small delta 22, all the MF capital delta 199 or 21, and even MF capital delta 200. Okay, so by now, I hope that you know the sources of cycling of metals in the environment had a direct impact on life. So up next, I will give a brief overview of the sources and the cycling of mercury, of metals. So first, the sources tracing. So the principle to distinguish natural source or anthropogenic source is to look at the isotopic composition, because both natural processes and anthropogenic processes would produce different fractionation and hence different isotopic composition in different sources, as you can see here from these three figures for copper, zinc, and mercury isotopes. So in geological history or areas remote from human industrial activities, natural sources would be the major contributors to the biogeochemical cycle of metals. So this is including volcanism or surface rising. We take the example of zinc emission from La Soufrière active volcano on the Guadeloupe Island. What we see using zinc isotopes, that the zinc in fumarole gas is generally derived from magma degassing and enriched in light isotopes. On the other side, zinc in hydrothermal solution is mainly derived from water-water interactions and enriched in heavy isotopes as a result of cold precipitation of sulfide or adsorption on the iron oxyhydroxide. So for natural Wiesing, the geochemical behaviors of metals can be assessed by examining the local Wiesing residue. And here I give you a first investigation of mercury isotopes during Wiesing processes. What we can see in this picture, so the only mass-dependent fractionation occurred with light isotopes proficiently released into solution, and this fractionation is mainly controlled by the species of mercury in solution and also the related adsorption. We can also even verify the result by studying another isotope systems. Here, in this case, we show you the isotope composition of gallium. So what we found is that in the same profile, gallium is partially mobile, with light isotopes enriched in Wiesing residue, and the fractionation is mainly controlled by the changes of aluminum or iron-related minerals. So once the isopolonic process comes into play, and like what is happening in today's world, metal isotope fractionation can be used to trace the emission sources and the historical evolution, as shown by, for example, the divergent trends of mercury and its isotopes. Especially since the industrial evolution, mercury emission display a clear increase together with significant changes of capital delta 199 and small delta 202. So from previous slide, we can conclude that metal isotopes provide reliable ways to trace their releases from both natural processes and human activities, and which may ultimately constrain life evolution. So next up is a brief view of cycling. So as far as cycling is concerned, one of the main issues on a global scale is the flux movement between, for example, atmosphere, terrestrial, and ocean environments. So metal isotopes, for example, here, mercury and zinc, can provide an independent way to constrain their cycling. For example, in the atmosphere, I have made an important observation of anomalous mass-independent fractionation of even mercury isotopes capital 200 in precipitation. So what we can see, the 200 anomaly show a clear seasonal variation with higher values in winter and lower values in summer. I just want to mention that this even MF observation was made from a rough pass, because as I mentioned, the mechanism for even MF remain unclear. So at the beginning, we found high values of 200 in our samples, and even in summer, it's still there. So what we think, damn, so samples probably be contaminated in the lab. So what we do, we process the two standards using exactly the same protocol for those precipitation, and we found no EMF at all. So it means probably even EMF do exist in natural samples. And you'll see in their figure that during the blanket period, I left Canada. So, and then what I do, I had to go back to Canada to collect 3 more winter samples. I found exactly the trend came back with high values in winter samples. So I want to mention also, though the mechanism remain unclear, the conservative behavior of even MF can help us also together with all the MF to revise the existing model of global mercury cycling. In fact, this type of fractionation is very sensitive. Just to give an example, mercury isotopic composition in precipitation of Guiyang City, southwest of China. And what we can see that almost all samples can be explained by the mixture of long-distance transport and the local anthropogenic emission. But there are 3 samples they weighted from other samples. So this is identified to be caused by Jost paper burning. Maybe you don't know, on the tomb swiping day, when Chinese people burn a lot of Jost paper for commemorating the ancestors. And we also performed a sample burning experiment in lab, show exactly what we found or determined in these 3 samples. Okay, the mass independent fractionation of even isotopes, even MF, is also successfully used to determine the entry mechanism of elemental mercury into a terrestrial ecosystem, for example, the plants. So as you can see in the figure, the plant samples to the green points processes almost identical capital delta 200 values as atmospheric elemental mercury, and very, very different from elemental mercury 2+. So this means, unlike those all other metals, their uptake mainly from the soils. The uptake of atmospheric elemental mercury through, for example, leaf stomata was identified as the dominant pathway of mercury into plants. Another example on the terrestrial cycling is tracing zinc in the Seine River, France. So we found using zinc isotopes that the dissolved zinc in the Seine River is mainly derived from anthropogenic input. Here, the plant treated with water, as you can see in the pictures. And I just want to mention that maybe and the preparation of 2024 Olympic Games in Paris had started, for example, in 2006, when you can see in the picture he's sampling the sign. And the result actually helped to, at least a certain extent, to guarantee the opening ceremony on the top and some aquatics on the Seine River on the left. Okay, multidimensional fractionation of mercury isotopes is particularly useful to tracing mercury cycle in the ocean. So these are another successful application. So what we can see on the left picture, that almost all ocean samples, including seawater, belt, and sediments, they display even MF values in between those of atmospheric elemental mercury on the left, and also atmospheric mercury 2+ values on the right. So this means that atmospheric elemental mercury is actually a major contributor to the ocean. This is a significant advance relative to the traditional model. And the traditional model showed that the main deposition of mercury is the atmospheric mercury 2+. So I want to mention that this result was obtained based on limited seawater mercury data. And because obtaining such data is extremely difficult due to the fact that the mercury concentration is very, very low, and the PPT even less than 1 PPT level in seawater, and also because of the interference is strong. So the good news is that we developed a new method to overcome these difficulties. This is achieved by precipitation of mercury as sulfide following the MCSCP mass measurement. So the method is suitable for very, very low mercury concentration, and especially convenient for on-site sampling handling on board. So this is very important. So in fact, the development of this method is a tough story. I show the picture. Now this work was done by my previous student, Yulong. And actually, and he was supposed to work in the government before his PhD. And after 2 hours of meeting with me, he decided to work in my lab. And he worked hard and spent several years, almost 5 years, to develop the method. Because the concentration is very, very low, we need even 15 liters of seawater to get one accurate measurement. So after the publication of the paper, and Yulong just went back to government again. So to sum up, the metal isotopes can trace their own trajectory, like we see, and then metal isotopes, especially multiple isotopes, can constrain complex biogeochemical processes and the related environmental conditions, for example, redox and climate changes. And by now, we can see that the isotopic geochemistry shows great power in tracing the sources and the cycling of metals in natural environment. So what we want to do to extend such success into biological system, particularly because the traditional stable isotopes have paved the way for the entry of metal isotopes. And just for the uptake, this study showed that the intracellular uptake of zinc by marine diatom preferentially accumulate light isotopes, and also the switch from higher to low affinity zinc transport pathways results in the changes of zinc isotopic fractionation with zinc concentration. And the isotopes are equally powerful in terrestrial ecosystems. Here, the example shows that the uptake of zinc from soil to plant, and also its translocation can largely fractionate zinc isotopes, with, for example, light zinc isotopes enriched in leaves, but a varied isotopic composition in different horizons. Okay, similar successful examples can be found also in mammals. Just to show a simple example, in the figure you can see zinc isotopes in tooth animals shows that very clearly increase from commivoles to herbivores. So as a result of their difference, we know that in herbs and plants, zinc dirt 66 zinc values are much higher than in meat. And the zinc isotopes in this case in fossils can thus preserve, for example, paleodietary information. And for accumulation, and the mercury isotope fractionation can track, for example, traffic transfer in marine food chain. Importantly, mass-independent fractionation of mercury isotopes in marine animals can record photodegradation of methyl mercury in seawater because trophic transfer doesn't produce any mass-independent fractionation. And thus, the mass-independent fractionation can use to track the sources of mercury in marine biota. For example, on the right side, the decrease of mercury, both the mass-dependent fractionation and the mass-independent fractionation in fish with depths reveal probably increasing contribution of methyl mercury from deep ocean. And our recent work confirmed that mercury isotopes in deep ocean biota may make the upper ocean fish, like you can see, that the trench biota has almost the same isotopic composition for either delta 22 or capital delta 199. So this means probably the dominant contribution of elemental or atmospheric mercury even reach in abyssal trench. So this also indicates also that the human emitted mercury can even reach deep ocean after other atmospheric transport and also their deposition. And finally, for metabolism, isotopes fractionate also during metabolism. So just to show an example, at the cellular scale, redox and functional changes significantly fractionate copper isotopes, which generally, for example, heavy copper isotopes tend to be enriched in either oxidized species or stronger chemical bonds, and which may result in larger variation of delta 65 copper in different organs and body scale. But on the other side, unlike copper, zinc isotope fractionation is mainly triggered by functional changes at the cellular scale. Also, it results also in large variation of delta 66 zinc antibody scale. So now I would like to finally present a few case studies showing how metal isotopes can tell us about the core evolution of Earth and life. First case is how metal as a nutrient stimulates life evolution, an example of zinc availability and the evolution of eukaryotes in 2.1 billion von civilian group. And the von civilian group was reported to record the first putative multicellular organisms. And what we can see from both pictures, that the prioritized fossils are characterized by high zinc content up to 800 ppm, but much lower delta 66 zinc values, minus, for example, 0.15. And compare to the surrounding rocks, for example, the pyrite with lower zinc concentration and higher delta 66 zinc values. So this difference was likely caused by metabolic zinc handling rather than the hydrothermal activities or sulfide precipitation. So this suggests that the zinc availability and the utilization may have stimulated the evolution of eukaryotes. The second case is the toxic stress of metal on the extension of life. An example on the end-Triassic mass extinction, we know that the volcanism is the dominant natural source of mercury. So the large-scale volcanic emission can input a large amount of mercury in ecosystems. Here, the isotopes reveal that mercury pollution emitted by volcanism stressed plants. For example, you can see in the photo the spore mutation, and even for millions of years after emission. And this provides that new evidence of mercury isotopes for metal-induced extinction. The third case is metal isotopes as hypothesis of volcanism for mass extinction. Here, I give you an example of mercury isotopes again. We know mercury isotopes can be used to trace, for example, organic and non-organic sources, and also local emission and also remote emission. This is because organic emission generally has no mass-independent fractionation. But if mercury transported for long distance in the atmosphere, the photoreduction can produce positive mass-independent fractionation in mercury 2+. Once this mercury 2+ deposited in the sediments, it can produce positive capital delta 199 in sediments. And on the other side, the terrestrial input generally has negative capital delta 199. This is the reason why mercury isotopes have been used in many studies to trace the organic impact on the mass extinction. So just the example of the end-Permian mass extinction, we can see in the figures, and the mercury and the copper spikes together with their isotopes in the South China terrestrial sections indicate probably direct contribution of nearby volcanism instead of Siberian trap, and suggests that the impact of local arc volcanism on this mass extinction. So another example is that metal isotopes can function as a process to indicate ocean redox changes leading to the extinction. And for example, mercury isotopes can track the photic zone euxenia, PZE, and this is a kill mechanism for mass extinction. And generally, in PZE condition, mercury would mainly bind to sulfur-contained ligands. And the transformation, especially the photoreduction of mercury-sulfur complex, would induce a negative odd MF, but a positive MDF. This is the signature totally different from any other biogeochemical processes. So this allows for specifically trace this PZE phenomenon. An example show, for example, the later Devonian mass extinction, we found 3 positives of PZE correspond very well to FF extension and also DCB extension, indicating probably the hydrogen sulfur poisoning might be the potential kill mechanism for this extension. To sum up again, and we can see that the uptake, accumulation, and the metabolism of metals by life trigger distinct isotopic functionations. And the stable isotopes are irreversible tracers for metal biogeochemical cycling and the environmental changes, providing fundamental insights into Earth and life called evolution. So the question is, what is the next adventure of metals? Before I end my talk, I would like to spend the last few minutes to talk about an exciting new field or potentially a new aspect of metal isotope geochemistry, the medical application of metal isotopes. So all the findings I presented so far has pointed to the new application. For example, calcium isotopes refer to bone diseases, iron isotopes refer to blood, copper and zinc isotopes for the cancers, and magnesium isotopes for diabetes. Just to give a simple example here, the selenium copper isotopes show higher sensitivity to track the progression of liver cirrhosis. For example, very lower than 65 copper values found for liver cirrhosis than the control values. The control values mean the healthy person. And even lower values for liver cirrhosis with ascites. And such success is not random. I want to mention that more studies confirmed successful application of copper isotopes to colon and breast cancers. So on the left figure, we can see, and the average trend represents a breast cancer patient. And the threshold of minus 0.35% refer to the average value of those control group, the healthy persons. So values of delta 65 copper lower than the threshold of minus 0.35 indicates that the patient is close to death. If this threshold holds also for colon cancer, the selenium delta 65 copper, we can see responds much faster than traditional biomarkers here. The example of CA 19.9 and or ACE, I don't know what they mean, but anyway, copper isotope could be a potential biomarker for diagnosis of severe diseases, for example, cancers. So for completing the medical application of metal isotopes, interdisciplinary method and the theory that they incorporate metal isotopes and molecular biology should be developed. And then most importantly, systematic characterization of metal isotope fractionation in biological processes and the cellular or molecular levels is key to advance the application of metal isotopes in life and the medical sciences. I just shared the picture I took several days ago. Okay, this is the end of my talk. Thank you very much for your attention. Thank you.
Thanks very much. Thank you. Thanks very much. Now we have time for questions. So who wants to start? There is a microphone in the middle there that you can use. So maybe I start myself with a question. I've been, I thought it was very interesting, all the things you showed about the mercury isotopes. And I was wondering whether one way or another that could be used as a tool to study all the archaeological sites when the people were extracting gold and, you know, in the distant past. Whether that could be useful or not. I'm just wondering.
I think there are several studies actually done by the group in Toulouse. They use mercury isotopes to trace the to measure, for example, the higher and nail mercury isotopes to trace their sources, and definitely can use that isotope signature to show that the mercury in the human body derived from the local mining activities. There's some.
Okay.
Yeah.
Okay. Yes? Maybe you should introduce yourself, give your name.
Hello, I am Heather Shrimpton from the University of Waterloo. Are you considering measuring any other types of non-traditional stable isotopes for these studies?
I'm sorry, can you repeat the question?
Are you considering measuring any other non-traditional stable isotopes for these studies?
Yes. In my lab, I have developed a method, for example, measure, I mentioned the gallium isotopes. And the gallium isotopes, I think it's useful to tracing surface Wisen processes. But the geochemical behavior of these elements remain unknown. And generally, we think it's related to, for example, aluminum and iron geochemistry, as shown in the Wisen profile. We also developed a method for tin isotopes and also antimony isotopes. And those two isotope systems are redox-sensible systems, so there are many applications, especially in, for example, redox tracing. And we can measure barium, calcium, lithium, and magnesium isotopes as well. Thank you.
Great talk, Jiubin. I'm really excited about your potential to be a medical doctor in future. So based on your knowledge, could you maybe commenting on what other potential traditional, non-traditional stable isotope can be used for earlier detection of like cancer or Alzheimer's, not just the existing one? Do you have any comments?
I think there are several systems that can be used to trace, to trace such kind of cancers or diseases. So I mentioned that copper and zinc also, and we do laboratory experiments showing, for example, copper isotopes are very sensitive to liver cancer, but zinc isotopes are sensitive to breast cancers. And I think also, for example, magnesium isotopes can be used to trace diabetes. I didn't show the picture. One study showing that during the progressing of diabetes, I think there are T1D, T2D types, and the concentration of magnesium didn't change a lot, but the isotopic composition decreases. So I think for now, zinc, copper, iron, and calcium, maybe others, but they need to be developed in the future. Okay.
Can you comment on cobalt and molybdenum?
I'm sorry.
Can you make a comment on cobalt, because that's in B12, and molybdenum?
The cobalt.
Yeah, cobalt.
So cobalt only one stable isotopes. So that's the problem. We can't measure its stable isotopic composition.
What about molybdenum?
It's the same question, the same problem. Aluminum also has only one stable isotopes. Yeah, that's why we try to develop the method for gallium isotopes to see maybe it's useful to look at the biogeochemical behavior of aluminum.
Thank you, Professor Chen. Last talk is amazing. Actually, I want to ask one question. When you use the meta-isotopes to track the progress of some diseases, do you measure the blood samples or the body tissue samples for this kind of analysis?
That's a technical question. Actually, all tissue samples we can measure, either blood or tissues and tumor and hairs, like biological samples, like leaf, like roots, we can measure in the lab. It's not a difficulty. The problem is, I think to get the samples, actually, it's very, very difficult to get true human samples for such kind of research. That's why we try to get right samples to do the experiment for example, for cancer diseases, and to see what happens for copper, zinc, iron isotopes during the progression of liver cancer, for example.
Thanks. Okay.
Hello, thank you for your talk. I'm Ekaterina Rojas from the University of South Carolina. I do work with molybdenum isotopes, but my question is more social. So how fast or what and how cheap is to do the analyses of the stable isotopes in blood, and how fast do you think it will become a common tracer that like hospitals could provide for patients of cancer? How far are we from real advance?
I think for zinc copper, it doesn't time consume, and we can measure like a natural geological samples. And the problem is, so we needed to digest samples, like all geological samples. We need to purify the samples to get pure zinc or copper from organic matrix, and then measure it. So generally, we can treat in my lab 20 to 30 samples per day, but use a different column, of course, and also pass the samples in the following days on MCSP mass. But for mercury, it's very, very difficult. I just want to mention. So, the mercury concentration, I didn't work on the human body. The natural water samples are very, very low. We need even filtrate, for example, 10 to 20, even 50 liters of water. It takes several days, even 1 week, and then pre-concentrating them. So that's why if you use a column, it takes also another week. So that's why we developed a method for precipitation of mercury from natural samples. It can be done in, for example, 1 day.
Thanks.
Yeah, thank you.
Okay, so if nobody wants to ask any more questions, we're going to congratulate Jiubin again for his beautiful lecture, and we had a great time, and thanks very much.